So it took a global pandemic to finally get me to make this video, so here we go. So this is the Respiratory and Digestive Exam Study Guide. It's really simple, which is why I haven't made a video for it, but for those that at least want to get walked through the study guide, I'm going to do it now. So the first part of that test is a vocabulary section. So in the study guide, you have different definitions for certain terms. All the terms are underlined. So on the test, you'll have the definition portion, and you will have a word bank with your selections for your different vocabulary terms. So for example, if the first question was actual site of gas exchange, then you'd find alveoli in your word bank and then put that letter that's in front of alveoli on your Scantron, so bubble in that selection. So first part, very simple, just like you used to do with vocabulary tests in elementary school, or not sure if you guys did them in middle school or high school or whatever, but at least I remember for elementary. So really simple. Just remember your vocabulary. Most of this stuff you already know. We've already covered for the respiratory stuff, where the alveoli are, our different respiratory volumes, knowing that the phrenic nerve is the one that innervates our diaphragm, knowing what tidal volume is. So here it says about a half a liter, which if you necessarily do the math, but half a liter is also the same as 500 milliliters. So a lot of this stuff you already know. Next part of the test is the alveolar ventilation rate calculation. It's only one math problem, it's multiplication. You also will have multiple choice answer selections for the actual math problem. So you don't actually have to really do the math if you're good enough at estimating. So no, you won't have a calculator. No, the equation will not be on the exam. And you will have to know for the exam that the air that actually reaches the alveoli is 350 milliliters per breath. So this is something we covered in the respiratory chapter. So with our tidal volume, our relaxed breathing volume of 500 milliliters, only 350 of those milliliters actually reach your alveoli. So the equation for the alveolar ventilation rate is the air that ventilates the alveoli, which is our 350 milliliters per breath, and you multiply that by the respiratory rate. So on the test, you'll be giving a different number for the respiratory rate. So in our example that we'll do below, we have a patient who is a 120-pound female, so just regular person. There is some more math stuff that goes into extra when you're getting specifically on estimating how much air is really reaching the alveoli for that person, and part of that is based on weight. But we're just going to use regular normal person, so we're just going to use the 350 milliliter estimation. So we take our 350 milliliters of breath, we multiply that by our respiratory rate of 16, which is also breaths per minute. So our breaths cancel out, and so we're just left with milliliters per minute as our final unit. And doing the math, we end up with 5,600. The respiratory rate, again, will be different for the exam. Yes, you'll have to know the 350 milliliters. You won't really actually have to know the units, because all of that will be a part of your answer choices. So long as you either can do your math or at least can estimate it close enough, you'll be fine for that one. Do note that one of the answer choices will be, instead of using the 350 milliliters per breath, using the tidal volume of 500 milliliters. So I forgot what I did for the other two other math choices, which are just like common math errors, but that is an option where instead of using the correct air that reaches the alveolar, I use the tidal volume as one of your incorrect answer choices. Okay, let's move on. So next part is labeling, and that one's pretty simple. So here's your picture. If you're on my on-campus class, you've already done this picture. Very similar to for your digestive exam for exam 8. So again, same concept. Where the parts are, labeling, very simple, easy to practice. I know some people, what they'll do, because this is a Word document and it is editable, I think. Oh, nope, never mind. It's not editable. That was a picture I put in there. Okay, well, they'll take this picture, and then they'll cover up the answer choices, and then they'll just practice using the picture. So putting everything where it is. So very simple, easy on labeling. Okay, second to last section has to deal with the questions about the Digestive Lab. Now, I know it didn't exactly work the way we would want it to, but this one is some basic questions that happened with the lab. So in this section, same thing, it'll still be a numbering. You'll still have a word bank with different letter choices for putting it in on the Scantron. You'll just be filling in the blanks. So for example, for this sentence, the Burette's reagent turns blank when polypeptides are present. So you choose from the word bank, whatever is pink, and put that letter into your answer choice, or for whatever that number is. So same thing with the rest of these. You're just filling in the blanks for stuff you've already gone through. So Benedict's, that was the aqua blue one that turned gold in the presence of glucose, even though currently the amylase enzyme we're still working on it because it's really strong, and so it ended up working before you guys even got to your lab bench. Oh well. Stuff works at 37 degrees, which is our body temperature. Pepsin likes a pH of 2. A sample that goes through all the steps of the reaction. So when we did the digestion lab, we didn't have controls for everything, but let's say if you're doing a drug study and you have some patients who have the placebo, which is usually some form of sugar pill or a non-active source of treatment. So it looks like you're doing the treatment, but you're actually not, and you mimic as much as possible the actual treatment. And then you have another group of patients that actually gets the treatment. The patients that didn't get the real treatment would be your control group. So they went through the same steps like the actual treated patients, but they didn't actually get real treatment. So this is what they'll do, is that's kind of like testing the placebo effect, as in, okay, how really well is this drug working? Is it mostly because of placebo that people are believing that it works, and so therefore they think that they're getting better, and whatever it is, their body's having a positive effect to be able to, work on healing itself to get better or whatever. So for example, in our digestive lab, for let's say the lipids one, where we had the test tube that had water in it, so it doesn't have the active portion that would necessarily help it with digestion. So your controls in this case would not work. There's other levels of control where you might have one that shouldn't work and one that should completely work, and yeah, it gets complicated. But for our digestion lab, go back to the lipids. Our control was the one that did not have bile, and then our experimental one is the one that did have bile. So in that case, we did have something where it went through the steps, but it didn't really have the active variable that's being tested, as in the presence of bile. The other one, so just, what goes on? So what enzymes are breaking down what? These two dealing with what happens with our detectors. So for example, phenol red, our pH indicator. Our phenol red is normally red when it is at a pH of 7, which is neutral, but then it turns closer towards a yellow, or for some of you guys, it was more of an orangish color, so still slightly more yellow, because it became more acidic. So just seeing the fatty acids build up in the solution, it became more acidic, and so the color changed. Here's another indicator, the iodine. So iodine is normally brown, but it will turn black when there is starch. So, we had issues with this for the lab, but you did see that it did turn that blue-black color when you placed it into the test tube. Okay, the last one second to last one. Forgot about that one. Okay, I'm sorry. This is the second to last one. This one's the last one. Okay. So for the catabolism reactions, again, this is just more stuff that you learned during the digestive chapter. So we have different nutrients. We got proteins, we got lipids, we got polysaccharides, so our carbohydrates. The proteins are broken down to polypeptides and then to amino acids. So these are the correct orders. Lipids broken down to emulsification droplets, which are then broken down into our fatty acids and our glycerol. And then our polysaccharides, which are our carbohydrates, broken down to disaccharide, so di meaning for 2, and then down into monosaccharide, so mono meaning 1. So for this part of the test, you will have the option of saying whether or not the order that is presented to you is correct or incorrect. So let's say hypothetically, let's go with the polysaccharide one. So let's say on this sentence, it says disaccharides are broken down to polysaccharides and then monosaccharides. So obviously that's the wrong answer, so the wrong order. So your answer on your Scantron would be incorrect, which would be your B. Okay, this one is the last one, I promise. So the last portion is the enzymes of the digestive system, what they're breaking down, what's their product after breaking down, what's the trigger for its production. So we have 3 different enzymes, even though bile isn't an enzyme, but whatever. So 3 different enzymes, and you're basically going to be filling out a chart. So you're going to have the numbers in the chart to place for your different enzymes. So for example, if pepsin, let's say that one's the first one, because I don't remember. Pepsin, let's say that's the first option. So your organ site for production, so you'd look in your word bank. That word bank, you'd have stomach as one of your options. Don't worry that some stuff does appear multiple times. It's fine the way it is. It just happened to work out that the letter choices end up being the same, so it doesn't really matter. So it's okay. And plus, the way it's coded on the Scantron, my little key, it's fine. Okay, so organ site for production. So we'd find stomach in our selection choices. Bam, put it for that number. The nutrient that it digests, so carb, protein, or lipid. So in this case, pepsin breaks down protein. The product after digestion is polypeptide. So find that answer in your word bank and you're good. The trigger for production is protein in the stomach, and so that one go in our last section. So for that last column, you'll have 2 different answers, but it's still fine. The way it works out, where it's like, okay, do I, which one should I put where? It's fine. It works out. It's okay. So all of the same thing for bile, you'll go through the same process with the organ, the nutrient, the product, and also with pancreatic lipase. Yeah, that's it. So real simple, very easy way to rack up a bunch of points, kind of stuff them in there. And yeah, that's it. So real simple. It's a lab exam. It's not supposed to be hard. So have fun studying.